Okay, so now let's talk a little bit more about the structure of the plasma membrane that allows some molecules to cross and others not to cross, which is what makes the plasma membrane selectively permeable. We have two types of transport for things to get across. One is passive transport and one is active transport. These are the broad categories of transport. There are several types that we're going to talk about of passive transport and several types of active transport. So this is where your worksheet kind of comes into play to help try for you to organize everything that we're going to go through in this lecture and this following lecture on active transport. So the big difference when we talk about passive and active transport is whether or not it requires energy. So generally speaking, when we talk about passive transport, that means that no energy input is required. We don't need to add any additional energy. When we talk about active transport, energy input is required, usually in the form of a molecule called ATP, but other molecules like GTP can function in that manner as well. Okay, so we're going to start first with passive transport. Again, no energy input. So we don't need a molecule like ATP or GTP to come in and in order for it to cross the plasma membrane. However, all molecules, it doesn't matter if we're talking about a glucose or an ion or anything, they have what's called intrinsic kinetic energy. So it's not outside energy, it's energy within the molecule itself that naturally makes it want to move in a specific direction. So under your worksheet where it says the process of simple diffusion, facilitated diffusion, and osmosis, the energy source is going to come from the intrinsic kinetic energy of the molecule. When we get to the forms of active transport, the energy source is going to be a molecule called ATP, which we'll talk more about at the end of this chapter. So again, we have three basic types of passive transport. We have simple diffusion, facilitated diffusion, and osmosis. If we're talking about simple diffusion, that means the molecule can directly cross that phospholipid bilayer. It doesn't need any assistance or anything to help get it across. If we're talking about facilitated diffusion, it can't directly cross the phospholipid bilayer. It needs to be facilitated, so it needs a protein to help get it across. And then when we talk about osmosis, osmosis just means that we're talking about the movement of water from an area of low solute concentration to high solute concentration. But we're going to talk a lot more about those now. Regardless, all the types of passive transport involve diffusion, which means we're moving something from a high concentration to a low concentration down a concentration gradient. And moving something from a high concentration gradient to a low concentration gradient means it's not going to require any outside energy or any energy input. So for example, if we're talking about diffusion and you take a dye pellet and put it in a beaker full of water, sorry about that. A dye pellet is extremely concentrated. If you just put the dye pellet into the beaker of water, it will naturally diffuse down its concentration gradient from the area of high concentration, which is the dye pellet, to the area of low concentration, which is all of the surrounding water. This naturally happens. We don't have to heat it up or provide any energy in order for it to naturally diffuse. And the whole purpose of diffusion from high to low is it continues to do that until we have an equilibrium. So it doesn't matter in this beaker if we were to take a sample from here or a sample from here, the concentration should be the same. So when we talk about how quickly can we get that something to diffuse, it's going to be influenced by three different factors. One is how much concentration do we have, the size of the molecules that we're trying to diffuse, and the temperature. So if we're talking about concentration, the greater difference in the concentration between the two areas, the faster the particles are going to diffuse. As far as size, the smaller the size of the molecule, the faster it's going to diffuse. And we're talking about temperature. If we increase the temperature, that's going to increase the kinetic energy within the molecules, which is going to increase the speed and therefore the rate of diffusion. So again, if we want to speed up the rate of diffusion, we would want to have a really high concentration difference between the two areas. We'd really want to have a small size of whatever particle or molecule we're trying to diffuse, and we would want to increase the temperature. So at some point we're going to reach equilibrium, and that's going to be where we have no net movement of molecules. There is never no movement of molecules because, like I mentioned, all molecules have what's called intrinsic kinetic energy, so they're constantly moving. It's just that molecules are moving in equal directions. There's no net movement. Okay, so remember the membrane is selectively permeable and only allows some things to cross. So when we're talking about lipid soluble, really small non-polar substances or molecules, those are going to cross the membrane via simple diffusion. It can squeeze between that phospholipid bilayer if they're really small, and if they're lipid soluble and they're nonpolar, that matches the inside of that phospholipid bilayer where those fatty acid tails are, so they can directly cross the plasma membrane. If they're a little bit larger and they're not lipid soluble or they're polar, they can cross the membrane, but they're going to need some help of some sort of protein, at which point we're going to have facilitated diffusion. And then osmosis, we're only talking about the movement of the water or solute, and I'll talk about that in a little bit because I don't want to confuse you right now. Okay, so let's start with simple diffusion. Again, small, non-polar, lipid-soluble, or hydrophobic substances, like something shown here, they can directly cross that phospholipid bilayer. They don't need the help of anything. Things that can do that or would be considered nonpolar and small are gases, so oxygen and carbon dioxide. They can enter and exit the cell via simple diffusion just by directly going through that bilayer. Also, steroid hormones, as we would expect, because in our plasma membrane we have those steroid structures already embedded in there, and fatty acids. Facilitated diffusion, again, these are things that are a little bit larger. They might have some hydrophobic areas. They're going to be needing facilitated diffusion, some sort of protein in order to allow it to cross. There are two types of facilitated diffusion. There is carrier-mediated and channel-mediated facilitated diffusion. If we're talking about carrier-mediated facilitated diffusion, that's where the substance of the molecule is going to bind a protein carrier. If we're talking about a channel-mediated facilitated diffusion, the substance is going to move through a channel that's typically filled with water. So let's look at the first type of facilitated diffusion, which is carrier-mediated. And these carriers that are going to help facilitate the movement of the molecule across the plasma membrane are integral proteins. So we can see they're embedded right here. And each carrier can transport a specific polar molecule or a specific class of polar molecules. So they are pretty specific. They don't just allow anything to cross. And what happens is when these molecules bind to this protein that's in the plasma membrane, it's going to cause it to change shape. So initially this is its shape right here. When something binds, it's going to cause it to change its shape to look like this, which allows that molecule to go from the extracellular fluid to the intracellular fluid. So basically it's going to allow for moving a molecule from one face of the membrane to the other. However, when we talk about carrier-mediated facilitated diffusion, there is a limited number of carriers present in the plasma membrane, so there's something that we call saturation, and that's when all of our carriers are engaged. So the molecule's transport is going to be occurring at its maximum rate when we have a saturation point for these carriers. The second type of facilitated diffusion is going to be the channel mediated. So this isn't going to change shape like we saw in the other one, the carrier mediated one. So channels are already embedded within the plasma membrane. Typically they have some sort of aqueous filled core, so they allow water to pass. And as water is passing, these other solute particles can go ahead and cross. So things like ions, I already talked about water, but they are allowed to cross along with whatever solvent or water that the cell is in. However, they do provide a little bit of specificity because the pore size, so this area right here, will not allow all molecules to cross, and same thing with charge because proteins are made up of amino acids, and amino acids have different charges depending on which amino acid we are talking about. We have two types of channel mediated facilitated diffusion proteins. We have leakage channels. These are ones that are always open and they allow ions to move via their concentration gradient. And we also have gated channels. So the opening of these channels is going to be controlled either by a chemical or some sort of electrical signal. The third type that we need to talk about is going to be osmosis. When we talk about osmosis, we're talking about the movement of water or the solvent, not the solute particles. So when we talked about facilitated diffusion or simple diffusion, we're talking about molecules crossing the plasma membrane. When we talk about osmosis, we're talking about the movement of water, either from the outside to inside of the cell or from the inside of the cell to the outside of the cell. So water can diffuse across plasma membranes. It can go directly through the phospholipid bilayer because it is small and polar. So sometimes they can cross directly through, which is shown right here. They can also go through what we call these specialized transmembrane proteins that are called aquaporins. So they're also allowed to cross via that route. So now let's talk a little bit about something called osmolarity. Osmolarity is going to be the concentration of the total number of solute particles in the solvent, and water concentration is going to vary with the number of solute particles, not just the type of particle. So when we talk about osmosis and the movement of water, water is always going to flow to the area where we have the highest number of solute particles. So for example, if we have a membrane right here that does not allow molecules to cross, and then right here, and we have a beaker like this. We have a high concentration of solutes over here. We have a low concentration of solutes on this side. If our membrane right here was permeable to these solutes, our solutes would naturally want to go to the left, to the side of lower concentration until they reach equilibrium. However, if we say that this membrane does not allow for these solute particles to cross, water is naturally going to flow to the area of higher concentration so that it can reach equilibrium. So until we get the same concentration of solids and water molecules on both sides. So here's an example. If we had a membrane that was freely permeable right here, so we can see we have a large opening for these solutes, what's going to happen is our solutes are naturally going to want to go from the high concentration over here to the area of low concentration over here. This doesn't require energy, so this is an example of passive transport. So they naturally want to go from an area of high concentration to an area of low concentration until they reach equilibrium where they have equal concentrations on both sides. However, if we're talking about a selectively permeable membrane, which is basically equivalent to our plasma membrane, and say we have unequal concentrations of solute particles on either side of this selectively permeable membrane. So our plasma membrane acts in the same manner. It doesn't allow these solute particles to cross. But water can cross the plasma membrane, or the selectively permeable membrane, either directly through the phospholipid bilayer or through those aquaporins. So that's exactly what's going to happen. So water moves to the area of higher solute concentration. So it would move in this direction. And again, the whole purpose is if we flood this side with water and increase the amount of water, we dilute it so that we have equal concentrations on both sides of the membrane. So that's how water moves. So this is just a little bit of an overview, not super important unless you're taking principles in biology, because we don't have cell walls like plants do. But when hydrostatic pressure equals osmotic pressure, that means that you're not going to have any further net movement of water. Hydrostatic pressure is the pressure exerted by water against the cell wall in a plant. If you're an animal cell, you do not have a cell wall, so we do not have hydrostatic pressure. Because of that, our cells can shrink or they can expand and get larger, which leads us to tonicity. So we don't have a cell wall, so we have the ability to change the shape of our cells. And a couple of our cells, or one of our cells in particular, are our erythrocytes, our red blood cells, that are capable of doing this based on the amount of water that they have on the inside. So if you take a red blood cell and you put it into an isotonic solution, which means it has the same number of solid particles out here, concentration of solute out here as it does inside, so it's the same, that's why it's called isotonic, nothing happens. You have your normal red blood cell shape. There's no net loss and there's no net gain of water. Water is still crossing the plasma membrane, but is not crossing it heavier in one direction. So isotonic solution, if you put a red blood cell in that, its shape stays the same, no net movement of water. However, if we take a red blood cell and we stick it into a hypertonic solution, hyper is going to have more. So for example, say we have like 1,000 solute particles out here. And a low solute concentration on the inside relative to the outside. Water is going to want to move to the area of higher concentration. So water is naturally going to flow from the inside of a red blood cell to the extracellular space to try to even out those concentrations. When it does this, it's going to cause the cell to shrink. And it can shrivel and die if it's placed in an extremely hypertonic solution. The third thing that can happen is that we take a red blood cell and we put it into a hypotonic solution. So the solution that's surrounding the red blood cell has a lower solute concentration than that of what's inside of the cell. So here's our red blood cell with tons of solute particles on the inside. And we put it into a hypotonic solution, which means it has a lower solute concentration. Water is always going to move to the area of higher solute concentration in order, again, to bring it to some sort of equilibrium. So water is going to move from the extracellular fluid to the inside of the cell until we have equal concentrations. And when this happens, it can cause the cell to swell or even lyse or burst open.